Experimental autoimmune encephalomyelitis, or EAE, is an animal model used to develop new and improved treatments for multiple sclerosis patients. Inducing disease in EAE requires a perfect oil in water emulsion to achieve consistent results. The method of emulsion preparation presented here is free from human influence, thereby limiting inconsistencies from one experiment to another. Even though the method has been optimized for EAE, it can also be used for other autoimmune disease models and also to test cancer vaccines.
To prepare the CFA slurry, add 100 milligrams of freeze-dried M. tuberculosis H37RA and 5 3.2 millimeters steel beads to a 7 milliliter tube. Shake the tube in the homogenizer for 60 seconds at the highest speed setting. Add 5 milliliters of IFA adjuvant to the tube and shake again for 60 seconds at the highest speed. Then, transfer the slurry to a fresh tube with a pipette, leaving the beads behind, and store at 4 degrees Celsius until use. To prepare a CFA peptide emulsion, download supplemental file 1 to calculate the amount needed for each reagent. Then, place the screw-capped tube and reagents to be used on ice. Next, add the emulsion components, first PBS, then the peptide, the CFA slurry, and finally IFA. Close the tube with the cap firmly by tightening and loosening it several times. Shake the tube vigorously for 5 to 10 seconds by hand to pre-mix the reagents. Place the tube in the shaking homogenizer and secure it with the rod. Set the speed to the highest setting and the time to 60 seconds. Once the run is finished, place the tube on ice for 3 minutes and repeat the run 1 or 2 more times with the same settings. Centrifuge the tube at 300 G for 1 minute to remove trapped air and compact the emulsion. Remove the cap from the tube, insert the plunger into the tube, and push it down slowly until it reaches the top of the emulsion. Remove the snap-off enclosure at the bottom of the tube by twisting it. Next, remove the plunger from all injection syringes and add a needle to the injection syringes. Then, attach the back end of the injection syringe to the dedicated lock at the bottom of the tube and lock it with a short twist. Transfer the emulsion from the tube to the injection syringe by pushing the plunger gently. Stop when the emulsion reaches the 0.15 milliliter graduation of the injection syringe. Separate the injection syringe from the tube and insert the plunger carefully, taking care that no air enters the syringe. Push the plunger until the emulsion comes out of the needle and repeat the process for the rest of the emulsion present in the tube. To analyze the size of the water and oil particles, add a tiny drop of the emulsion to a microscope slide, smear it out with a cover slip, and then push hard in a circular motion to flatten out the emulsion. Examine the smeared emulsion under a phase contrast microscope with 400 times magnification and focus on a field with a monolayer of the emulsion. Ensure that small uniform gray or white particles are visible. Emulsions produced by the homogenization method displayed gray or white bean-shaped particles, representing tiny droplets of water and oil emulsions. In contrast, the emulsion droplets produced using the syringe method were larger and whiter. In the vortex method, large gray or black particles were observed corresponding to air bubbles trapped in the emulsion. Changes in particle size distribution over time, measured using a laser diffraction particle size analyzer for water and oil emulsions, did not show any change in particle size over a period of 6 weeks. The fresh 3-week-old and 15-week-old emulsions all induced similar EAE diseases in all animals tested throughout the experiment. Mice developed a similar disease pattern in all experiments, revealing that the homogenization method presented here produced emulsions that induced EAE in a consistent and reproducible manner.
Before making the emulsion for an experiment, download supplemental file number 1 to calculate the amount of each component needed to minimize waste. The method presented here could be used to standardize emulsion preparation not only for preclinical research, but also in the future to prepare cancer vaccines in the clinic.